Hello everyone, my name is Visalhounuo. In this video, I'll be explaining on oxidative fermentative tests. The oxidative fermentative test was developed by Hook and Lifson in 1953. They developed OF media to differentiate between oxidative bacteria that produces acid from carbohydrates under aerobic conditions only, and fermentative bacteria that produces acid both under aerobic and anaerobic conditions. Carbohydrates are organic molecules that contains carbon, hydrogen, and oxygen in the ratio CH2O, and organisms use carbohydrate differently depending upon the enzyme complement. The pattern of fermentation is characteristic of certain species, genera, or groups of organisms, and for this reason, this property has been extensively used as method for biochemical differentiation of microbes. Saccharolytic microorganisms degrade glucose either fermentatively or oxidatively. The end products of fermentation are relatively strong mixed acid that can be detected in a conventional fermentation test medium. However, the acid formed in oxidative degradation of glucose are extremely weak and less, and the more sensitive oxidation fermentation medium of Hook and Lifson OF medium is required for the detection. The medium was made by increasing the amount of glucose above that found in medium used to detect fermentation and by decreasing the amount of peptone. The OF medium of Hook and Lifson differs carbohydrate fermentation media as follows. The concentration of agar is decreased to 2% from 3%, making it semi-solid in consistency. This assists in the determination the motility of the organism. Second is the concentration of peptone is decreased from 11 to 2%, decreasing the amount of alkaline products produced by the metabolism of peptone, thus reducing the neutralizing effect of these products. Third is carbohydrate concentration is increased by 0.5% to 1%. The increased concentration of glucose in the medium enhances the production of this weak acid to a level that can be detected by bromothionyl blue indicator. Objective of this test is to detect the oxidation or fermentation of carbohydrates by bacteria. Principle of the test is the oxidative fermentative test determines if certain gram-negative rods metabolize glucose by fermentation or aerobic respiration oxidatively. During the anaerobic process of fermentation, pyruvate is converted to a variety of mixed acid depending on the type of fermentation. The high concentration of acid produced during fermentation will turn the bromothionyl blue indicator in OF media from green to yellow in the presence or absence of oxygen. Certain non-fermenting gram-negative bacteria metabolize glucose using aerobic respiration and therefore only produce a small amount of weak acid during glycolysis and Krebs cycle. The decreased amount of peptone and increased amount of glucose facilitates the detection of weak acid thus produced. Dipotassium phosphate buffer is added to further promote acid detection. Requirements of the test are liquid paraffin, bacterial suspension, inoculating loop, spirit lamp, incubator, and 2 tubes of Hook-Lifson media. Coming to the procedure, sterilize the inoculating loop on a spirit lamp, then dip it inside the bacterial suspension and inoculate into 2 tubes of Hook-Lifson media. After that, pour the paraffin on top of it on one tube to make it anaerobic, and then keep both the tubes inside the incubator and incubate it for about 24 hours. The next day when you come, you have to observe for color change in the tube and observation. If it is positive, a positive carbohydrate utilization test is negated by the development of a yellow color in the medium. If it is oxidative, you will see a development of yellow coloration in the open tube only. If it is fermentative, you will see a development of yellow coloration in both open and closed tubes. And then if it is negative, you will see that the carbohydrate utilization test is indicated by the absence of yellow color, media remaining green or turned blue, non-oxidizer or non-fermenter. Result interpretation. Fermentative result will be indicated when acid production on both open and closed tubes. The acid produced changes the pH indicator, bromothiamyl blue, from green to yellow. Example, Escherichia coli. Oxidative result will be interpreted when acid production in the open tube, which is aerobic, and not the oil-covered tube, which is anaerobic culture, indicates an oxidative result. Non-fermenting bacteria that metabolize glucose via oxidative metabolism give an oxidative result. Example, Pseudomonas aeruginosa. Non-saccharolytic or negative OF test will be interpreted when non-saccharolytic bacteria give a negative OF result. The negative result is indicated by no color change in the oil-covered tube and in the same cases as increase in pH 7.6, changing the bromothiamyl blue from green to blue in the top of the open tube. The increase in pH is due to amine production by the bacteria that break down the peptone in the medium. Example, Alkaligens fecalis. Control of the test is, in case of glucose fermenter, is Escherichia coli. Glucose oxidizer is Pseudomonas aeruginosa. Non-saccharolytic is Moraxilla species. Uses of this test is, OF test is used to determine if gram-negative bacteria metabolize carbohydrates oxidatively by fermentation or are non-saccharolytic, have no ability to use the carbohydrate in the media. The limitation of this test are slow-growing organisms may not produce results for several days. Second is, some microorganisms do not grow in the OF medium. It may be necessary to use another basal medium containing dextrose to confirm the negative reaction.